Okay, this is for period, whatever was last, E, F, G. Okay, so we're going to finish the little part of the lecture we were working on here. So you should get to this point where we were talking about the DNA template on one side. This is just like you made at the beginning of class, and the DNA template on the other side. So we left off talking about the pairing rules, G and C pairing together, and C and G pairing together here. Step out, 3 minutes. All right, a couple things that you need to know. Same deal happens here. We get a bond between the phosphate and the number 3 carbon. How do I know it's number 3? All right, our AP chemistry friend in the back told me that this is the 3 carbon here, this is the 5, this is the 3, this is the 5, okay? They're the same sugars. Now, I'm gonna finish up the rest of these, so there's gonna be 2 more nucleotides. So there'll be a phosphate, a sugar, and a base, and a phosphate, and a sugar, and a base. Our base here is going to be a 2-ring base. 2 rings. This is going to be base A, because it matches with T. Here we're going to have a 1-ring base. It's going to be T, that matches with A. Once again, same deal. They're stuck with this bond between the phosphate and the number 3 carbon. We end up with a 3 end, or a 3' end, and down here, we end up with a 5 end. All right? The importance behind this is that DNA is parallel, side by side, but it's also, they call it anti-parallel, meaning they go in opposite directions, kind of like driving on the road. So far so good. Here's where the difference is. The difference is these are all covalent bonds on the other side. Every line that you see here, this line, all those are covalent bonds. Okay. The difference is what's going on between them. So between the bases, there are small, light interactions between these bases. We'll draw them as dotted lines. These small, light interactions are not strong bonds at all. If you think back to when we talked about bonding, the weakest of all bonds are hydrogen bonds. So in the center here, we have hydrogen bonds. And the idea of these hydrogen bonds is that these are super easy to break, which is important for your DNA to do what it needs to do. For example, there are times you need to break apart your DNA or unzip your DNA, unzip the genes that are there. And what happens is it opens up these bases to the environment. It allows you to copy it, allows you to make RNA from it, so on and so forth. There were a couple other things I wanted you to see as well. I don't think we're going to quite get to it on this next test. DNA is always a double strand like this. So there's one strand here, one strand here. From a mechanical standpoint, the outside part on each side is called the backbone. All right? This is what secures the bases in place, this backbone. And the backbone is made of the phosphate and the sugar. Same deal over here. The backbone is made of the phosphate and the sugar. In the middle here, this section in the middle, G's, C's, A's, and T's, this is the code. The code that makes you are the order, the A's, G's, C's, and T's in the middle. Okay? To give you an example, I'm throwing a rough number out here, but every cell, one single cell in your body has somewhere in the neighborhood of of 60 trillion bases in one cell. All right, body's made of millions of cells. Okay, actually billions of cells. So the number of bases that you have is just astronomical. We talked a little bit earlier is that if we look at the DNA, the DNA looks like a ladder. The 2 sides of the ladder are your phosphate and your sugar. And the things in the middle of the ladder, or the rungs of the ladder, the steps of the ladder that are in the middle, these are the bases. The bases make up the code. The code sequence is super important. When we look at humans and chimps, the code is almost 99% the same. We talk about human to human, it's even closer. We talk about people in the same family, it's even closer. We talk about twins, it's almost identical, depending on the type of twin you are. All right? So the code is very important. This code here in the middle This code here gives us Mr. tRNA, okay? All the code does is code for RNA. That's it, nothing else. Mr. tRNA is small. The reason it's small, it's able to leave the nucleus, go out into the cytoplasm of the cell, then you can turn that Mr. tRNA into proteins, which is the only thing you basically do, is you're a big sack of proteins. You take the DNA, you make RNA, those RNAs make proteins for you, the proteins do stuff for you. Helps you maintain body temperature, grow, make hair, make new cells. Anything that happens inside the human body is controlled by the genetic code of the DNA. If we work backwards and we talk about these small mRNAs making proteins, we can look at how proteins are similar. For example, blood types, A, B, O, AB blood types. If you have A blood type, all people with A blood type make the same end protein because they had the same RNA, because they had the same genetic code, okay? When you have mutations there in really important proteins, like the proteins found in your blood, you then tend to start having problems, okay? I mean, we all have mutations. I use myself as an example. I think the young ladies to the left also have the same problem. All three of us are lactose intolerant. Why? Because our genetic code had a change somewhere. That change gave us a different message, which gives us a different protein. And that protein's the wrong shape to digest lactose, which is a sugar. All right? How do you fix it? Well, you can take lactose pills, right, which is a supplement for the protein. Or you can go back and find the mistake in your genetic code and fix that. But with 60 trillion bases, super hard to do and super scary. All right, we're going to stop here. I hope this helped. I'll do another little quick video later about some of the things on the test.